Hey everyone, welcome back. In this video, I want to go over a really important molecule for life, and that is DNA. You've learned about DNA before. Most of you guys will tell me when we first start talking about this in class that DNA is the molecule that makes you, you. It's what makes us all different from each other. And you're right, it is. It's more of an instruction manual than anything, though. It's not the thing that really makes us, it's the instructions to make us. And so in this video, what I want to do is go over how DNA is put together. So we're going to learn the structure of this molecule first, and then we'll go into more detail about how DNA really is an instruction manual, what makes it a good molecule to hold all of the genetic instructions for us. So let's break the name down really quickly. DNA stands for deoxyribonucleic acid. So really it's two words here. Deoxyribonucleic is one, and then acid is the second word there. So that's what DNA means. That's what it stands for. Deoxyribo is actually talking about the sugar that's found in this molecule here, and nucleic acid is talking about the type of biomolecule or large macromolecule that DNA actually is. And again, it is just the instructions to make every protein that makes you up. So that's what we mean when we say that it's the genetic information for you. It's the instruction manual to make your proteins. If we look at this model right here, this is a really detailed model of DNA. It shows all of the atoms and the bonds that hold the atoms together that make the molecule DNA. We're going to go into some detail, probably not this much detail, but we will talk about all of the parts of a molecule in this video. So let's go ahead and dive right in. It would be silly to talk about the structure of DNA without talking about some of the scientists that are credited with coming up with or with understanding the structure of DNA. And so in 1953, these two gentlemen, James Watson and Francis Crick, both published a work that showed us what the structure of DNA really is. These two guys were able to come up with this model based on the work of many, many other scientists and understanding of chemical bonding properties, the way certain things will bond together and hold together. And then through some work and pictures taken by, I did put Rosalind Franklin here and Maurice Wilkins because these two were involved in actually helping get some of our first photographs of DNA here through what's a method called X-ray diffraction. And so you can see one of the images that they got here of DNA, and what they were able to show with this, what this helped Francis and Crick deduce, is that DNA is actually a molecule that's twisted in shape. And so we knew that there were certain elements and certain molecules that were there because of some tests that have been done on DNA. We've known about DNA for a very long time now, but the structure of it wasn't really known until the work of these people here. So let's take a look at the large structure of it first, and then we'll kind of dive into it in more detail. So the first part, when we look at DNA as a big molecule, got tongue-tied there. We refer to it as a double helix. This twisted molecule over here that's kind of hypnotizing you. It's actually making me a little bit dizzy. DNA doesn't do that in the nucleus of your cells. This is just an image there to kind of show you that it is a twisted molecule. It's a long, twisty rope laddery type thingy. But really, when we talk about it being a double helix, there's two sides to a DNA molecule, and each one of these two sides is going to twist. And so it's double-sided, and it's helical in shape. So then that's where we come up with a double helix there. So I'm going to move this DNA thing is making me dizzy. Let's move on. So when we stop it from spinning, and when we flatten it out and get rid of the helical twisted shape to it, we can look at the two sides in a little more detail. It looks like a ladder, and so you'll hear DNA referred to as a twisted ladder or a double ladder or double helix. It does look like a ladder with sides here and then steps or rungs in the middle. Really, I want you to recognize in this image here that there are two discrete sides to DNA. And so we have this side here, and then we have another side over here, and they're actually held together in the middle by hydrogen bonds. We'll go over that again, but I want you to know that these hydrogen bonds in the middle hold the two sides together. This picture over here gives us some names and a little bit more detail to it. It shows the hydrogen bonds in the middle. Some of these parts, these rungs, these nitrogen bases that make up the steps, some of them will form 2 hydrogen bonds, some of them form 3. And so the steps of our ladder are made up of what are called nitrogen bases. And then the sides, or the rails of our ladder, are made of alternating sugars. This D is deoxyribose sugar, and phosphate molecules. So the sugars and phosphates are bonded together, holding the sides or the rails of the ladder together. And then our nitrogen bases in the middle will bond together to make the steps. So those are our 2 sides, sugars and phosphates going down the rails, and then the steps are the nitrogen bases. Diving down even further, when we look at this molecule here, these things that make up the parts of our ladder can be broken down into discrete nucleotide molecules. Now, if you've looked into biochemistry and you've talked about the 4 big biomolecules that are needed for life, deoxyribonucleic acid falls into the category of a nucleic acid molecule. Well, nucleic acids are what we refer to as polymers. They're really large molecules that are made up of smaller repeating subunits called monomers. In the case of a nucleic acid, the polymer of a nucleic acid, the monomer that makes that is a nucleotide. And so as a nucleic acid, DNA is made up of a bunch of nucleotides that are strung together, and this is a picture here of what those nucleotides are. Each nucleotide has 3 pieces that make it up, so we can break that down even further. The first piece here is a phosphate group, and then we have a 5-carbon sugar that makes these up, and then a nitrogenous base. So these are the 3 parts of a nucleotide: a phosphate, sugar, and a nitrogen base. With DNA, and I said at the beginning of the video, DNA is named after the sugar that's in it. This 5-carbon sugar in DNA is what we call deoxyribose. In another video later on, we'll learn about RNA. And so RNA actually has ribose in it. It's a different sugar. Really, the only difference is that ribose has an extra oxygen. Deoxyribose has been deoxied. It's lost an oxygen. But in DNA, the 5-carbon sugar that we see is deoxyribose. And so if we were to test a molecule, if we knew that it was a nucleic acid, we could determine if it was DNA or RNA by really looking to see if it had deoxyribose in it. So again, phosphate, sugar, and nitrogen base are what make up the nucleotides here. Now, when we look at that even closer, our nitrogen bases here can be one of 4 different kinds in DNA. We have adenine, which is denoted here with an A. We have thymine, which is denoted with a T. And then over here we have guanine, which is a G, and cytosine, which is C. And so with DNA, we could have one of these 4 nitrogen bases. Now, the sugars over here and the phosphates are going to be the same. On every nucleotide that you see with DNA, the sugar will be deoxyribose. The phosphate group will always be the same. But the nitrogen bases can be different. So there are 4 different nitrogen bases in DNA, and they are adenine, thymine, cytosine, and guanine. Now, when we look at those, they have specific bases that they will pair with. And when we look here, I put A and T in the same color because adenine will always, always, always bond with thymine in DNA. Thymine will always bond with adenine. It's what we call a nitrogen base pair. These are the base pairing rules. So A and T go together, and then C and G go together. Years and years ago, I had a student that came to me one day after we had first learned about DNA, and he said, "Randolph, I figured it out, man. Apple tree chewing gum." And I thought, what is this kid talking about? This doesn't even make sense. He says, no, it's just the way DNA bonds together. A and T go together, so apple tree, and then C and G go together, chewing gum. And I thought, well, okay. It doesn't make any sense, but it does kind of have a catchy ring to it, so apple tree, chewing gum is something that I've taught kids for years now, and it just seems to stick in your head. And if you can remember that, with DNA, A and T go together, and C and G go together. Those are your base pairing rules. Now, when we break those down, when we look at our nitrogen bases, we can classify those into 2 different groups themselves. Oh, looks like my computer wants to change something. Let's close that for now. We can break our nitrogen bases into 2 categories. We have what are called purines, which are structures that really have 2 rings to them. So we can see one ring here and then a second ring here. Our purines are going to be a larger nitrogen base, and those are adenine and guanine. So adenine and guanine are purines. And then pyrimidines are a single ring structure, thymine and cytosine. And it, a little trick to help you remember that is I always thought of pyramids as being huge, but in this case it's kind of the opposite. The pyramids are the smaller ones, the pyrimidines. I know it's silly and it's a simplified way to remember it, but it worked for me when I was in school. And so if you can remember, the adenine and guanine are the purines, and then thymine and cytosine are pyrimidines. Why this is important is because when we look at the bonding of this, and like I was saying with Watson and Crick, they knew that the way certain molecules bonded together helped determine the structure of this big molecule. Our purines are always going to bond with a pyrimidine. And so here we see adenine is going to bond here with thymine. And what's even more special about this is there's going to be 2 hydrogen bonds formed between these 2 molecules here. So the adenine purine bonds with the thymine pyrimidine. And then down here we have the guanine purine is going to bond with our cytosine pyrimidine. And with these, there's going to be 3 hydrogen bonds formed. These hydrogen bonds are weak and they can be broken pretty easily, which is going to come into play when we talk about DNA replication and then how DNA is actually the instructions for our genes. We talk about transcription later on in another video. But I just want you to know that adenine and guanine are our purines. They are bonded with thymine, and cytosine are pyrimidines. And between these we have 2 hydrogen bonds, and here we have 3. So that's how that works. Those are our base pairing rules. The last thing that I want to talk about here is what we refer to as the anti-parallel nature of DNA. And so when we break that word apart, anti-parallel, we're talking about something that creates parallel lines. And so our 2 strands of DNA here are parallel lines. They run side by side. They never cross over each other. They do twist, but they don't cross over each other. And so they are parallel lines, but the one thing about it, the thing that makes them anti-parallel, is that this strand of DNA is going in this direction, and this strand of DNA is going in this direction. So they're actually going in opposite directions. And the reason we say that, going in opposite directions, has everything to do with the structure of the nucleotide and then the way that these things the way the DNA molecule gets read by different enzymes. So let's kind of break that down. What I've done here is I've shown you there's a 5' end here and a 3' end here. That's what when you see the 3 with the apostrophe here, we call that 3' and 5'. Now over here, we have 3' and 5'. They're opposite. So where I have a 5' end on this side, I have a 3' end on this side. And it has everything to do with the structure of the nucleotide. So let's look at that really quick. Here, I can see that when we talk about nucleic acids, we're going to number the carbons in this molecule. Well, everywhere there's this little angle here, this is a carbon. So there's a carbon here, there's a carbon here, carbon here, carbon here, and then our 5th carbon here. And when we do this, we always start clockwise from where the oxygen is in this sugar molecule. So 1 prime carbon, 2', 3', 4', and 5'. Now, when we break this down and look at our individual nucleotides, where does the phosphate group attach on this deoxyribose molecule? Well, it attaches to the 5' carbon. So if I'm looking at a strand of DNA and I see the end of it has a phosphate group hanging off, that's going to be my 5' end. That's what I'm going to refer to that as. And then the end that doesn't have a phosphate group attached, the bottom side of this deoxyribose is the 3' side. And so that's my 3' end. So when I look at this, the way that this gets put together, I have a 5' end and I have a 3' end. And if I follow this all the way down, it only makes sense that this will be my 3' end. And again, following this down, that will be my 5' end. So there you have it, the structure of DNA. I hope that helps clear some things up, and we'll see you again in another video.